Okay, well, thanks everyone for waiting. We'll get underway. First of all, I'd like to, on behalf of the ARC Centre of Excellence for Plant Success in Nature and Agriculture, I'd like to acknowledge the traditional owners and the custodianship of the lands on which we meet. We pay respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society, and of course, we recognise the many different traditional owners and different lands across which we meet, which is in a virtual meeting very, very diverse. My name's Christine Beveridge, and I'm the director of this centre. And it's a genuine pleasure to introduce Rana Munns. Professor Rana Munns is recognised internationally for her research on fundamental principles of crop adaptation to salinity. And she's always been very passionate, I think, about, doing things that are useful, figuring out how to make the science useful and has a much greater impact on science generally than perhaps many of her colleagues, because she's really interested in people, in advancing plant science as a discipline, recognition of the importance of plants, and then the people themselves, the plant scientists. In terms of her core research, she's extremely well known worldwide for her work on salt tolerance. She's discovered genes for sodium exclusion and led a team identifying genetic basis of salt tolerance in durum wheat, and has had impact to the industry with increasing yields in farmers' fields. She's retired from CSIRO Agriculture and Food, and we're very fortunate that she continues, though, to interact with us still at the bench. I think living at Lennox Head in New South Wales, a very nice place of the world. She has an emeritus professor position at University of Western Australia, a fellow of the Australian Academy of Science and World Academy of Sciences, has many accolades to her name. Rana, look, it's a real pleasure to have you speak to us today. I believe you're going to be talking about, different capacities plants have in their arsenal to adapt to abiotic stress. And we've asked Rana to give a sort of general, recognising that we have a fairly general audience. So, Rana, thanks so much for your agreeing to sort of step out of your busy retirement schedule to both prepare and then deliver this presentation.
Well, hello everyone. Thanks very much to Christine to invite me to give this talk. I don't give talks very often these days, so it took me just a little bit to get in the hang of it, but I enjoyed preparing this talk. It's based on a paper that we wrote the end of last year. I wrote with Harvey Miller, also from UWA, and was invited by the journal Experimental Botany for an issue they're doing on centenary reviews. And I know Christine has already written a review for that same. So I think could I move on to the first slide? Next slide, thanks. Yeah. So it's about abiotic stresses in general, not any one in particular. We're looking for common factors that plants have and use to adapt to stresses in general. So just to introduce, I considered that we do have two existential threats. We have the threat of climate change and also the challenge of feeding an increasing world population, when we know actually that the best agricultural land is being reduced in area by expansion of cities, if nothing else. So climate change causes increased global temperatures and reduced crop yield, and then the world population demands more crops and pastures on decreasing traditional land. And next slide, thanks, Phoebe. Now there's a couple of quotes from the latest IPPC report of last year, which was pretty comprehensive, like 1,000 pages, and they've summarised food and vegetation production in the whole of the world, and they're looking at the problem of decreasing land for productive use for farming and grazing and so on, and illustrated here, a very present threat is sea level rise. Not only will it flood, or it is flooding lowland areas, countries like Bangladesh and West Africa, which are very low lying, but with climate change you have greater chance of much more severe storms. So when the storms are whipping up oceanic waves, they're then intruding onto coastal land and up rivers. And one place that we'll talk about a couple of times is Bangladesh, when all these coastal storms are forcing seawater up the river, and it's almost now reaching the capital city, Dhaka. And of course, it takes a long time to drain away because the land is so flat. Next slide. So this is in southern Bangladesh, just illustrating how flat it is. And this is one of the local farmers. Their husbands have gone to the cities to try and earn some money from a job, and then the women and children run the farms. And there has been a lot of work on this, and on the lady's right is a new salt-tolerant crop that's been bred by the Bangladeshi Research Institute, just empirical breeding, really, and is really supporting farmers on this land subject to seawater intrusions. Next slide. And this is just a clip out of their book that's showing the major constraints they've picked out are the soil nutrients, heat stress, aridity. I was surprised to see ozone there, because I thought we'd sort of stopped worrying about ozone now, but apparently in Northern Hemisphere it still is a problem. Is it exhaust from cars, I guess, things like that. Another thing that I haven't thought about so much is the pests and diseases, and this is by far the greatest impact of climate change, according to this, is the increase in pests and diseases, mainly in the Northern Hemisphere. I guess we don't see that so much. Again, I guess it's mainly the temperature that has caused the increase in disease stress on crops. Next slide. So the idea of this project was publications that have been around in the cancer literature for some time. Now, cancer is a very widespread and highly researched phenomenon. There'd be millions and millions of cancer researchers all over the world. They are very specialised. So you have one group, looking for all the different cancers, all publishing their own papers, all going to their own meetings, very rarely talking to each other. And this stimulated Hanahan and Weinberg to try and put it all together, to try and draw out the commonalities of what is causing a cell to become cancerous. And he wrote a paper 20 years ago called The Hallmarks of Cancer. And I thought 3 of these 6 apply pretty much to crops. So the first thing is, cancerous cell, apoptosis is disengaged, so it stops dying. It produces its own growth signals. So instead of relying on growth signals coming from neighbouring cells or in the bloodstream, the cancer cell produces its own blood signal. And so what's the other major thing that's happened is the cell develops a resistance to growth suppressors. So normally, the outside tissues are regulating growth by suppressing growth. The cancer cell develops a resistance to external growth suppressors, and then it produces its own growth signals and hormones, and then this at the bottom enables replicative immortality. So this idea of these things will apply to every single type of cancer, and it was published in Cell, and it has got 40,000 citations. And I was just noticing the other day, they published another one 10 years later called The Hallmarks of Cancer, an update. And this, in fact, is the diagram from the updated paper, and that had 60,000 citations. It went down pretty well in the cancer, the fraternities. Next slide, thanks. So what we did, sort of put together an illustration based on that idea that we picked out, this Harvey and I picked out 7 capacities. I mean, it could be more, could be less, just depends how you sort of divide them up. But 7 capacities, and these in rotation, say, starting from energy, which is in the petrol pump, and repair, because those 2 things go together, and then communication. We've summarised that as this is what you have. And then we talk about supply chain, carbohydrates going down to roots, nutrients coming up from roots, and selective uptake from the soil as exclusion. So that's moving essential nutrients and products up and down the plant, and then how a stress cell can cope with, can change to grow in a better position by asset management, which is basically root to shoot structures and shape shifting and developmental changes. So that's what we're proposing. It really is what a plant under abiotic stress is called on to do. Next slide. Now, more recently, there was a paper published by this ecologist, Leslie Head from Melbourne University, and Catherine Phillips, I believe, is at UQ. And in a ecological sense, they suggested that there were 5 or 6 plant capacities used by weeds to invade. So we have these capacities, and almost by corollary, you could say that any plant with all these capacities is likely to be very invasive. And the capacities they highlighted were movement, which is more like like twining, that sort of a movement, sensing, communicating, and flexibility of form, which allows them to grow in a different type of, say, more shady or more sunny environment, and the reproductive timing, so that sort of sped up as well. So I thought, well, that was another idea of having capacities which is in common to a lot of plant species in a lot of environments. Next. And then we thought that this was a useful thing to follow for a number of reasons, which I find very frustrating. You've got all those omics studies, and whether it's transcriptomics or proteomics or metabolomics, all throwing up hundreds of genes or metabolites that are either upregulated or downregulated under stress. And honestly, there's very few of those metabolites or genes that have really proven to be useful. You know, papers just publish these lists and they sit there. I think the same criticism could be made of a lot of the GWAS studies, that they'll bring up perhaps only tens or hundreds of genes, but then, you know, the researcher has to select the one that might be really important and then do further studies with it. And this is rarely done as well. These studies just sort of sit there. So I thought if people thought about capacities, you could help distinguish between the primary and secondary responses. So you could chuck up, you know, 90% of the genes that change that are really just in response to what we call housekeeping genes. And it could, just a personal thing, discourage studies exploring transcription factors with no known function. I think the transcription factors function should be seen in one of these capacities. Next. So these are what we decided to focus on. Pretty much includes everything a plant does or can do, really. And I'll just now go through them briefly, one by one. Next. So the first obvious thing a plant does under stress is, in almost every case, shoot growth is reduced and root growth is not. The roots either maintain their normal growth or they actually increase, particularly because they're under nutrient stress. When the plant under nitrogen or phosphorus stress, the roots continue to proliferate, presumably to search for more N or P. In the meantime, the shoot growth is reduced because it can't be supported. And then, because under drought and salinity stress, in particular, shoot growth or leaf evaporative area is reduced to maintain enough water to continue growing. And in some cases, senescence of leaves is delayed, and in fact the senescence of old leaves is increased because old leaves are just a cost to the plant. Old leaves, they're shaded, and while they're there, they're just chewing up carbohydrates. So an adapted plant will be continuing to produce younger leaves and sloughing off its older leaves. Next. Something we call shape shifting, which is often called shoot architecture or root architecture. That always changes under stress. Leaf morphology will change. The surface of the leaf will change, like thicker cuticles, say, to reduce heat load. Rooting depth to explore for fresh water at depth. The shoot apex will change in structure from vegetative to reproductive. That's usually hastened under stress. So we've got signals coming from roots or shoots that would initiate the change in development from vegetative into reproductive structures. And that's probably the most important capacity of all to respond to stress. And this is just typical of on the left is a plant treated with salt. The same thing would happen if it was in a drying soil, that, okay, the leaves are smaller, but there's fewer tillers, but the leaves are thicker. The specific leaf area is less, so you've got less surface, thicker leaves, and therefore the chloroplasts are denser and the leaves look a darker green. Stressed leaves always look a darker green, and they often look blue because of all the wax that has developed in the cuticle. Next. Now, the supply chain I won't say much about because it's pretty obvious to people like us that the transport in the phloem from leaves to shoots has to be maintained, and in balance, the supply of water and nutrients from roots to shoots has to be maintained. And again, this would rely on a change in the allocation of structural assets. Next. And this was so why I asked Harvey Miller to help me write this paper, because he's spent a lot of time looking at energy generation, respiration, and so on, and repair. That in the leaf, most of the proteins turn over daily. Certainly Rubisco does, some very quickly, and he's calculated this probably consumes about a quarter of the ATP generated by respiration. You've also got the turnover of membrane lipids, and then there's also the balance between what carbohydrate is used for energy and then what is put aside, say a starch, for storage. That's another drain on the plant. So next, communication, obviously long distance hormonal signals, and there's all sorts of signaling pathways within cells and between cells. And I won't talk any more about that because you guys know a lot more about it than I do. Next, and then selective uptake capacity, which has been more in my area of research, and got membrane transporters that are induced when the nutrients in the soil are low. Nutrient deficiency will induce nitrate and phosphorus transporters. And also salinity, you've got transporters that can select for potassium over sodium, and also within the plant lining the transpiration stream, they can actually remove sodium and not potassium from the transpiration stream. So next. So that just covers all the capacities that we considered enable plants to respond to abiotic stress. And I mean, all plants have them. It's just how much they are used, or they're able to use. Very small genetic differences make a big difference between the ability of the plants to actually enact these capacities. Just a little aside here, we thought we should, for us anyway, define stress as an external stress, not an internal one. So a lot of people say that plants are stressed. Well, that could mean anything really. It could mean, oh, poor Diddham's little plant is stressed, It's almost a psychological term. So we want to be very clear by stress, we're talking about environmental stresses. And so in the use of the physical sciences, which refers to a force per unit area or pressure. So stress is the external force, and then the strain is the response. So in our paper, we've always talked about responses of plants. In physical sciences, strain is a change in the shape or volume or length caused by the stress, like, material that's bent a little bit. It can damage or compress or cause a catastrophic failure. So that's quite a good analogy between how a plant responds. And then in the chemical and biological sciences, the chemical sciences, the strain is a change in the energy status of molecules or the plasticity of form of the structure. So I think we're used to the word plasticity in response. And again, we're using it here regarding a plant, a single plant. We're not using it in an evolutionary sense, as ecologists would do. And next, the reason we're very careful about that is that there's a phrase called oxidative stress, which is used all the time by almost everybody, and I think means, well, it's very confusing. I think it doesn't mean very much, and it's really, I think, created a lot of easy way outs. We just say, oh, the plant's under oxidative stress. And how do you know? Well, because you measure the reactive oxygen species, or you measure an antioxidant, and they've gone up. Therefore, the plant's under stress. And then there's a big guess about whether these changes are beneficial or not, and whether they should be selected for or not. And sometimes they're just used as a surrogate for growth. So people don't bother measuring whether a plant's growing well or growing not so well, because that's fairly difficult to set up. Well, it's very tedious and expensive to set up a glasshouse experiment and measure plant growth. It's much easier to pick a leaf and measure the reaction, measure an oxidized oxygen atom or an antioxidant or something. That is just so easy. And that's taken over as a surrogate for growth. So just wanted to point out, 20 years ago, oxidative stress was due to UVB radiation or pollutants like ozone and sulfur dioxide. Now it's mainly used in the opposite way. It refers to what's happening inside the plant. We're suggesting actually this so-called oxidative stress is a response to the external stress, has changed the metabolic activity and growth rate. And it doesn't mean any more than that, really. You can't say it's a sign that the system is under strain, because RAS change all the time. They change as tissues age, they change diurnally, they tend to be high during the day, and they go down at night, mainly because of the light environment. So it's actually very difficult to select leaves at the right stage of growth to actually measure if there have been changes in ROS due to the stress. As Harvey pointed out, the only way you could tell whether there is oxidative stress is to measure the damage to macromolecules. So that means a measurement of precipitated proteins, peroxidized lipids, or oxidized DNA. And the easiest one, of course, is the measuring peroxidized lipids by the what's usually called the MVA, the mevalonate reaction, or also called T-bars. It's malondialdehyde, MDA, is often measured by people, and that tells you whether that's the final stage of lipid peroxidation, and that tells you whether there has been damage or not the level of antioxidants or ROS. And I would add one more point, that even if the NDA levels have increased, it still doesn't mean that the plant has been damaged by the stress, because it still could be growing quite well. It still could be putting out new leaves. So just measuring what's happened in an old leaf, I don't think really tells you whether that plant has adapted or tolerate the stress or not. So that's my little rant and rave. Next slide, thanks. So these are the stresses that we considered, and I'm not going into them in much detail, but these would probably be the main things that you think of when you think of abiotic stress. And in all these cases, you would be thinking it'd be nice to be able to choose a gene, or at least a genotype, that might be able to be used in breeding for abiotic stress, which we know is increasing in severity. Next. So yeah, please don't read all this. But what we did is we've lined up the various stresses on the left-hand side, and then we've lined up the various capacities at the top. And we've just given a few suggestions as to how these capacities are useful, in fact crucial, for adaptation to these stresses. And like the first one I've already mentioned, that in drought, the main thing you've got to do is decrease the leaf area to reduce the transpiration. And then that's connected with the supply chain of sugars balanced with water from the soil and so on. So I'm not going to go into each of these in detail because we haven't got time for that. But it was a useful exercise to see if we really felt that those capacities would be important in abiotic stress adaptation. And let me give you just a couple of examples. Next. So I'll give you an example for saline stress, which has been my area of work for some time. So just start saying that they're, so we've got the symbol for selective uptake or exclusion. Do not enter. So we know that these are the major control points for, say, sodium transport. So the sodium is not taken up from the soil. Water's taken up, but not sodium. That's the first point of exclusion. Plant excludes 95% of the salt while taking up water. So that's its main adaptation. Plants can modify sodium travelling in the transpiration stream from soil. And then there is a limited ability when the plant's in the shoot to be able to remove it again. Next. Oh, so keep going with this. And one more. Thank you. Oh, sorry, back. Sorry, I didn't know. Thanks. So this illustrates the same sort of point again, and we selected, knowing that that was the physiological basis of how to control sodium uptake, and knowing that was important, we screened a whole lot of genotypes, we found differences, we did all the crosses, we did the genetics, and working mainly with Evan Salaguda, molecular geneticist, we were able to identify 2 genes that control the movement of sodium from soil to leaves. The first one we called NAX1 for sodium exclusion, and it unloads sodium from the xylem and in the leaf base. And then with the second one, we call next 2, which unloads sodium from the xylem in the roots only. It doesn't work in the shoot. And this is the one that we've gone on to work with plant breeders with. And I just want to say at this point, this is all in durum wheat, but this next 2 is exactly homologous with the gene in rice, was called, the QDL was called saltol. And in fact, we got onto it from their work because in rice, we could use the gene sequences in rice and find the syntonous region in durum wheat. And this saltol rice has been used by IRI breeders for quite some time now, probably 10 years. And there are quite a few varieties that they've got out in the field with this saltol gene, which has improved the growth in saline soil. Next. And so this was our work. These were field trials with durum wheat that was, we've crossed with the source of the next genes that we found. And so we had durum wheat with and without next genes. And as a farmer at Moree had offered his field that he knew was rather saline. And then this is Richard James taking samples from the field. And then the next slide shows the grain yield coming from lines with the next 2 gene here. So the lines were all in the tamaroid, the durum background, and in a saline field, in a wet season, there was no difference in yield between the lines with the next 2, which was good because we had to make sure that they yielded equally well on good soil. But then when we went to block 3, which was the saline soil, lines without next genes were really reduced in yield, and lines with the next genes had a much higher yield. So in fact, lines with the next genes were 25% greater than lines without the next genes. So that's what we wanted. No effect in a good soil, but 25% increase in yield on a saline soil. So this material now, we gave that to all the Australian wheat breeders, AGT and all those wheat breeders. So they've been crossing into their current lines. We couldn't release tamaroid because it was superseded by that time. The wheat breeders say they have much better durum wheat, so we had to cross it into. Next. And this is the last stage of the story. So Richard James had crossed our next genes into bread wheat as well as durum wheat, and that had a lot more interest around the world. And some of the Bangladeshi wheat breeders showed an interest in it, and Richard got up an ACR project, which has been running now for nearly 5 years. And the Bangladeshi wheat breeders have taken our material, crossed it into their lines. And this is one of their trials on different sites. And yeah, it's looking good. The lines with our next genes are really increasing yield on the more saline sites. It doesn't make much difference in the less saline sites, but in the heavily saline sites, the next genes are crossing. So this is Richard was able to go to Bangladesh in February because of COVID, that was out for about 3 years. But anyway, eventually he got back, and the Bangladeshi breeders have been doing all the work, but Richard was there to appreciate the interest and the effort, and all the villagers turned out to look at the trials as well. So next. So this will be the last the last example. This is for flooding tolerance in rice. Again, at Erie, the physiologists and breeders have been working hard to introgress it's a transcription factor called sub-1 for submergence tolerance. And this was a trial done about, at least 5 years ago. So very successfully showing that rice with the sub-1 gene in the same background, IR64, was able to survive submergence and to regrow after the flood subsided. And that sub-1 gene is also being used now in breeding. There's a lot of varieties released by Erie that have the sub-1 gene, both for Philippines and India, I know. The sub-1 gene, it's a transcription factor. I'm just making a note of it. It represses gibberic acid synthesis, but it also represses GA signaling pathways, with the result that shoot elongation slows down, so the plant doesn't grow very much when it's underwater or half underwater. Growth slows, therefore it doesn't burn up the carbohydrate that it had produced when it was above the water, and that means the energy then is conserved for a lasting survival, so they can last about 2 weeks underwater and in this condition without growing, without depleting all their carbohydrate reserves. And then when the flood does subside, there's enough energy left for the plant to start growing again. So I thought that illustrates 3 of our capacities, the energy, the communication one, and the supply chain one. Next. Oh, and the last one. This work been done in Canberra by James Hunt and Richard Richards. And Richard Richards have been very keen to breed erect leaves, upright leaves, because most wheat looks like the picture in the middle, big strappy leaves, which is great because there's lots of water around. But if the soil dries out, and particularly if there's a heat wave, those strappy leaves get burnt, as you can see in the right-hand slide. Whereas the idea of these erect leaves is that they're vertical to the light. They don't capture so much heat either, as well as light. And Richard Richards, so the rather unlovely looking erect morphology of leaves is really much better suited to conditions in which are getting drier and hotter. So the next, so that's, I've just got a few comments at the end. Yeah, just a couple of comments. We think it's really good to have, if you're going to be doing omics or GWAS and so on, it'd be really good if people had some idea of what they wanted to find, what they expected to find, rather than just sort of saying, it's discovered genes. I think it's really good to have a framework to put these studies into, and it would be really good to have a hypothesis, but at least to have an expectation of what might be changing in the plant, and And also this would, if you're going to measure a phenotype, this would help you to decide what phenotype you're going to measure. I think if you've got a gene of unknown function, or a gene that's upregulated tenfold under a particular stress, it'd be really good to think about where it might fit into a metabolic pathway, which you can get from proteomics, or think about it in a physiological process. And I think this way, the omics studies might become more useful. Next. Yeah, sorry, I'm just, that was a bit repetitive. I would say all these omics studies reveal about at least 100, probably 500 genes that are upregulated or downregulated. What ones are you going to, decide to work further on? Next. Yes, so this is the last. Yep. Just talking about the still the problem with GMO products, and I know gene editing is the big thing at the moment, and there are a limited number of crops in which that is possible, and a limited number of countries which are prepared to accept them. I guess that will change over time. It has to, really, as the demand for food and, food, even the demand for pasture plants for feeding animals. I think that's the way you're going to get GMO material out there. But in the meantime, I think conventional wisdom means that you select for plants that do well and you cross them into current varieties. And so basically it's a trait-based selection system, but you need molecular markers in order to make it useful for breeders. So I just say I think it'd be, lovely to put on your new genes or whatever into one of these traits, which comes out of one of these capabilities. And I think we might be moving faster on a useful way to improve salt tolerance or abiotic stress tolerance in crop plants. And yeah, just acknowledgements. Thanks. That was the field. That was one about that trial sites at Moree, and the emus came in one afternoon, so that was an extra problem. But anyway, I think they got chewed out all right, and we did have a successful trial after all of that. And yeah, so thanks particularly to my young colleague Richard James, who did quite a lot of this work, and he's still carrying on with it. So thank you all for listening.
Thank you, Rana. I love that photo with the emus in the field. That's fantastic. Yeah, look, that was a really lovely overview of I guess adaptive traits, aren't they, that come together to collectively to enhance a plant's response to stress. But I really appreciated your taking the time to distinguish between that the stress being an external thing, whereas strain being an internal thing. I think that's really helpful. And I think helpful for lots of us to think about that role of, like you're talking about oxidative stress, and yeah, you're right about the papers. Many papers talking about oxidative stress when it's something the plant's generated for itself. Like, you're stressing yourself on purpose or what? So that was a really interesting and helpful reflection on that. That's certainly going to change the way I think about oxidative stress, which is great. I think also it was really interesting that you talked about 25% of the ATP is used for protein turnover in a given day. I think that's some of us spend a fair bit of energy in the centre thinking about energy, about supply and demand, and we probably don't sort of factor in a huge amount in terms of how much energy is actually spent just breathing, just respiring, just maintaining the status quo of the cells and so on. So are there any we can ask people to put questions in the chat if they've got any questions. Greg Grabatsky says, great talk, Rana, thank you. And Demilola Atwalogan also saying thank you, Rana. A few other people too, so that's good. If there are any questions, so oh yeah, I was thinking about the if I can ask a question. Measuring the MDA, it sounds like that's a tip that we need to pick up on for future work. Do you know anything about how that technique actually works in the lab? Is it difficult or?
No, I think it's fairly straightforward. Yeah, it is. It's often called T-bars.
T-bars.
No, yeah, T-bars. Don't ask me now what that that stands for. Yeah, I've got the method. I've actually not done it myself, but I don't do anything myself these days.
I know the feeling, Rana.
But no, I think it's very straightforward. And yeah, it's such a shame because people, they'll measure, sod and, peroxidase or even reactive oxygen species. And there's just, little things you stick under a microscope. But I think the MBA is really quite a simple and absolutely unambiguous thing to do. That's the good thing about it. Yeah, it just shows damage to lipids, lipid peroxidation. No, I'll dig that out.
Well, it's a good example that you raised because often auxiliary buds anything that's dormant is often said to be under stressed conditions, right? But I mean, I don't know, when I'm sleeping, I'm generally stressed. And let's not think of plants as humans, but there's more to it. As you said, there's so much more to it than defining the stress by the amount of reactive oxygen species because it doesn't make a lot of sense. There's a question here in the chat relating to reducing, that was something you mentioned, that reducing plant height, you talked about reducing plant height to kind of overcome water.
Oh, yeah.
And so on, excessive water. So this question was about whether reducing plant height is important for salt tolerance in forage and pasture species.
I think what's important is the evaporative area. So you just don't want to lose any more water than you can, but at the same time you want to carry out photosynthesis. So I think it's not the height so much, it's the leaf area that goes with it. So you can have quite a good height as long as your leaves were fairly erect. Otherwise, I think most plants reduce their leaf area by actually reducing the height, or the length of the leaf is reduced a little, but the width is reduced enormously. So the leaves are narrower. And that's, I think, all to do with just reducing the surface area to volume ratio.
Yeah, okay, thank you. And that was from Surendra Bhattare. So Pushpa would like to ask a question about the fact that in a natural system or even in a field system, there might be multiple stresses like heat, water, salt, I don't know, pests. Has your article, or have you thought much about what framework we should use to think about multiple stresses at the same time?
Well, we don't know. Actually, I just wrote a little review with, well, there's a book coming out which looks at multiple stresses, multiple abiotic stresses. Look, it's so difficult to do an experiment on multiple stresses. I mean, just think about it. You can't do an experiment combining drought and salt. Well, you can do a drought, but how do you know what the drought's caused versus what the salt has caused? Yeah, you can do multiple stresses, but to try and dissect them as to which stress has had more or less impact on the plant's response is really very hard. And you can't have heat stress without drought. And I don't think you can have drought without heat stress either in the field. They just go together. But no, how to quantify the responses to different stresses when they're combined, it's beyond me.
It's challenging. Okay, so the answer to that,
challenging, that's a word.
It's very, very challenging.
I won't say it's impossible, but it's challenging.
Yeah. So Graham's asked a question, which is one that the centre asks a lot of itself, and so I'm going to jump to that one next. And it really is about what are the most adaptive traits. So we talk about, you've touched on the idea of should we start at the bottom, look at the genes and how they affect the phenotype, or should we spend more of our energy thinking about how important each phenotype is for the success itself? And so he's sort of asking the question, should we go phenotype down more in our research rather than mechanism up? It's a philosophical question.
Okay, it's a philosophical question. I know. I think you've got to think. Look, you've got to think about both ways. But I'm not sure I could say which was the best. But as long as you're thinking about the top down and the bottom up.
I guess what we're asking is, if you've got a certain amount of resources at your disposal as a scientist or whatever, what's your sense of where you should put your resources? Should we be spending more time thinking about how all the phenotypes connect with each other and respond to lead to plant success? Or, so that's sort of physiology and phenotype, or should we be spending more of our energy on identifying individual genes and the mechanism at the bottom? And of course you have to do both, but if you were in charge of this centre, for example, where would you put your resources? Do you think we've finished at the top down already, or do you think we're just at the beginning?
Well, you can talk to a plant breeder. I mean, they're the ones that produce the results. They don't actually have a phenotype, but it's not just yield. I mean, they don't go just on yield. They always record flowering time. They'll record height. Of course, obviously diseases and things. And then we've now got these sophisticated phenotyping measurements. So that's actually one way of looking at it, that you've just got, really good automated phenotyping, and that you just select the plant with the great, it's grain yield and so on.
Yeah.
And then you keep crossing with it, and eventually working with a geneticist, you could probably come down to the gene level.
Well, this is exactly the thought we're having. Is it is when you've got a phenotype that works, then you're already onto something. And understanding the gene that underlies the phenotype going that way is actually very powerful, because you already know the phenotype has value. Whereas when you go the other way, you don't really know. Yeah.
Yeah, I think you're just working in the dark when you're starting from, well, so that genomics is not really going to get anywhere on its own, is it? That you're starting from transcriptomics, you really don't know where you're heading. You don't even know what you expect to find.
Yeah, no, agreed. Look, we're really running out of time. I really want to ask Anthony's question. He's asking about whether plants can actually have any cognition, or whether it's just a rearranging of biochemical pathways due to availability of resources. Do you think plants have got any cognition, Rana?
You're the one that you're best to answer that. I personally don't think so, but then I don't know anything about it. So.
Yeah, I think they've just got amazing networks of interacting components that are more like a network, and that when you bring a signal into that or a change to that network, it responds in a very predictable way. It's so sophisticated and fancy that it seems like it can think. But it's actually the outcome of the millennia of natural selection. Anyway, look, thank you so much, Rana. So people are asking if they'd like a recording. Will that be okay, Rana, if we hold a recording of this so that people who've registered can get a copy?
Yeah, that's okay. I think that's fine. I'd rather give you this paper when it's accepted.
Yeah. So when the paper comes out, Rana, we'll advertise it in our newsletter as a paper that people might like to read, because it's clearly a great topic and great relevance. So we'll do that. Plus we do have a YouTube channel, but yeah, we'd like to encourage everybody to read your paper when it comes out, which will when is that issue, Rana? It's
look, I'm guessing it's the middle of the year.
Sometime in the middle of the year, so only a few more months and it'll be out.
I think it'll be a couple of months. Yeah. I think so.
Yeah. So thanks for lingering an extra 5 minutes to have a further discussion. I appreciate that, and thanks very much for your time, Rana.
Oh, it's a pleasure. Thanks for asking.
Yeah, very welcome. Thank you.